All right, so now we're going to talk about classification of enzymes. I did research on enzymes for what was my entire scientific career before I started teaching. This was my least favorite subject to have to teach because it's like memorization of vocab. Somehow I was able to make it fairly successfully in my career without having had all these things perfectly memorized. Um, so this was something that I was actually tempted to cut out to not make you guys have to memorize it. And then I looked at the ACS exam and just found multiple examples of questions coming from this particular chapter. So not my favorite, a lot of memorization and vocab words, but unfortunately it is something we have to know to be successful on the ACS exam. Okay, so this is about classification of enzymes. Basically, each enzyme will fall into a particular category, and our goal is to be able to categorize these enzymes based on their function. Okay, and also pick apart a little bit of information from the name of enzymes. That's going to be the goal of this lecture. So first of all, enzyme names always end in ase. Okay, so sucrase is an example of an enzyme name. Again, you can tell because it has that ase ending. Some of them are named for the reactions, some enzymes are named for the substrates that they're catalyzing the reaction of. So sucrase catalyzes the reactions of sucrose, okay? Others are done by the type of reaction that they catalyze. So an oxidase catalyzes an oxidation reaction. And other ones just have common names, such as pepsin and trypsin. Those tend to be the digestive enzymes, which were some of the first ones that were discovered. And so they're kind of given their own fancy name that's just stuck around ever since. Those common names are the ones that kind of existed before this formation of this enzyme commission of the International Union of Biochemistry and Molecular Biology. Long story short, much like in organic chemistry, how we have an IUPAC nomenclature system for naming organic chemicals, at one point, a group of scientists got together and decided we should really have a standardized way for naming enzymes. That way, when I'm talking about the enzymes in my organism and you're talking about the enzymes in your organism, we can more easily see the overlap. Okay. All right. So we're going to talk about classes of enzymes and then examples of reaction of those enzymes that fall into that class and the type of reactions that they catalyze. All right. So first class is the Oxido Reductase enzymes. All right, and oxido reductases catalyze oxidation and reduction reactions in the cell. The two examples of this class of enzyme that we're going to talk about are oxidases, and those catalyze oxidation reactions, or dehydrogenases, and they can catalyze either oxidation or reduction reactions. I don't know why they named it like that. It should seem like anything that does an oxidation reaction should be called an oxidase, but here's an example. Alcohol dehydrogenase is going to catalyze the reaction from ethanol to ethanal, which is actually an oxidation reaction. But it's so a dehydrogenase catalyzes the removal or addition of two hydrogen atoms and utilizes a coenzyme. So a coenzyme that's often used in oxidation reduction reactions is this NAD to NADH. These are called coenzymes, or also sort of cofactors. These are things that are commonly used in the cell as a pair for another type of reaction. So alcohol dehydrogenation in this case is catalyzing an oxidation reaction because it is removing two hydrogens from this particular substrate, which is an oxidation reaction. Okay, so an oxidase or a dehydrogenase are both oxido reductase enzymes. Another one is a transferase, is another class of enzyme. Okay, and a transferase, also kind of nice because it's in the name, it transfers a functional group from one compound to another. So for example, alanine transammonase transfers an amine group from alanine over to it would be a glutamate. Okay. So yeah, so transaminase is going to be a transferase class of enzyme. Nice that, it's always nice when the names are similar. It makes it kind of easier. I will say that a kinase, so we talked about the transaminases, transfer amino groups, anything with trans in the name is going to be a transferase. Another really important transferase is a kinase. These are very, and again, it's going to be harder to kind of remember this one because the name's not similar, but it is really important to remember they are an incredibly large group of enzymes. They transfer phosphate groups from one substrate to another, and that turns out to be just a really important function within the cell. So a kinase is another transferase. We just got to know that. All right, a hydrolase is an enzyme that catalyzes hydrolysis reactions. So these are reactions where you're breaking a bond, but specifically water is being consumed in that bond breaking process. And this actually happens quite frequently. So peptide bonds are hydrolyzed, and that's how you break a peptide bond. Likewise, lipids, that ester bond in lipids is hydrolyzed to form that fatty acid and glycerol components. Sugars are the diester, the disaccharide linkage, polysaccharide linkages, those are hydrolyzed to break those. So all of these important classes of different biochemicals that we've learned about are broken up through hydrolysis reactions. So the hydrolase reactions are really important type of reaction where you're using water in order to break a bond. So again, protease is one that's going to break a peptide bond. A lipase is one that's going to catalyze the hydrolysis of the ester bonds in lipids. And a nuclease, we actually really haven't talked about DNA or RNA yet, but that's an example of there's a phosphoester bond in nucleic acids that's broken up by a nuclease. So all of these are examples of hydrolases which promote hydrolysis reactions, catalyze hydrolysis. Okay. All right, ligases, these are my least favorite class because I feel like it's, I don't know, for whatever reason, it's the hardest for me to remember, but these catalyze addition or removal of groups without hydrolysis. So they're basically making and breaking bonds without the use of water, okay? So a decarboxylase, so this is an example of converting pyruvate to ethanol. This would be a decarboxylase reaction where we're removing CO2. And notice that this bond is being broken, but it's not being hydrolyzed. It's just being broken, okay? So this would be an example of a decarboxylase removing a CO2. A deammonase, so removing an amino group. A dehydratase or a hydratase, these are adding or removing water from a substrate. So the classics, and I know that it's like, wait a minute, I thought it was without a hydrolysis, right? So these are going to be like either addition reactions. So for example, going from an alkene to an alcohol, right? This would be what we would have called in organic chemistry a hydration reaction. This would be catalyzed by a hydratase, and this would fall under that ligase group. Okay, or the reverse reaction would be a dehydratase. So hydratase catalyzed, dehydratase catalyzed, the removal of that water group from a substrate. Okay. Isomerases, these are reactions. So the key word here being isomer that you can pick from the name of this class of enzymes here. So an isomerase is one that catalyzes a rearrangement of a substrate, turning it from one isomer to another. Okay. otherwise known as isomerization reactions. Okay, but the key word there being isomer. So malate isomerase will convert malate to an isomer form of it, which is called fumarate. We don't even know what malate or fumarate is, so like don't worry if those don't ring a bell. Importantly, though, is what's happening to our functional groups about our double bond. Remember, double bonds are rigid, so these molecules are distinct from one another. They are isomers of one another. And so, yeah, that is what an isomerase is doing. They have names that end in isomerase, so that's always nice. It's always convenient when there's some continuity. Another one that's going to be a little bit harder to remember is an epimerase, which catalyzes a rearrangement of a carbohydrate. So if we remember a sugar, what makes galactose distinct from glucose is just the orientation of those OH groups on that carbon backbone. So an epimerase is one that will swap and create another sugar. So it could convert, for example, galactose to glucose or vice versa. Those would be isomerase classes of enzymes. And when you're specifically talking about a carbohydrate, it's an epimerase. All right, and the last one is what's called a ligase. Okay, and this catalyzes the joining of two substrates using ATP as energy. Okay, so ATP is something that we're going to talk a lot about when we're talking about metabolism. It is a common source of energy within the cell. It's used to help catalyze reactions that would otherwise not be very favorable. So for example, adding CO2, creating this carboxylate group. Let me use a different color. So we're going to add CO2 to pyruvate, creating a carboxylate group. That's not a favorable reaction, so we need to use ATP in order to make it happen. Okay, so when two molecules are joined together to create a larger substrate using ATP, it's called a ligase. So ligase linking together. These have names like synthetase, right? Because again, we're creating larger molecules, so we're synthesizing larger molecules. Or a carboxylase, which is what we just saw here. So taking CO2 from the gas and adding it to a particular organic compound is not a very favorable thing to do. So it needs CO2, but that is a carboxylase reaction. Okay, so all of these are ligases. Again, when you think ligase, you want to think linking stuff together. All right, so let's now use these to try to fill in the blanks here. And actually, first, what we want to do, I would suggest, is try to remember what the broader class is for all of these different enzymes, because these are all specific group or specific examples from what were the larger classes. What I'll say is, of course, when you're doing this, you're going to be using this cheat sheet a lot, right? This is a nice little set of, what am I trying to say? These tables are really helpful. They will make answering these types of problems a lot easier, and that's great. But of course, we're not going to have this table for the exam. We're not going to have it for the ACS exam. So we really need to make sure that we have a good understanding that you can do these types of problems without having to refer back to your cheat sheet. So of course, at first you're going to have to, because these are just so foreign sounding, but you want to get to a point using flashcards and stuff where you don't have to cheat. Okay. So an ammonase, so let's look at do we have an ammonase, a dehydrogenase, an isomerase, and a synthetase? Okay. Probably the easiest one to think of the broader class for is going to be this isomerase, because the broader class is also called an isomerase. Okay, these are the ones that catalyze converting a substrate to a product where the product is just a different isomer. Okay, the dehydrogenase. So looking back, which class is that going to do? Let's see if we can't just pick it out from the name. Dehydrogenase, this was this example, a dehydrogenase. We saw the example of alcohol. Dehydrogenase catalyzes either oxidation or reduction reactions. So this is an oxido reductase class of enzyme. Okay, a synthetase. What would a synthetase be doing? Would be kind of synthesizing or creating new bonds, linking stuff together. So this would be an example of a ligase. Right, a synthetase was an example of a ligase type of enzyme. Okay. And then lastly, an ammonase. So this one would be hard because if we looked back through our examples, there actually isn't one that has an ammonase as part of its example reaction catalyzed, right? But if we look here, a deammonase would catalyze the removal of ammonia from a substrate. So an ammonase is going to do the exact opposite. It's going to catalyze adding an ammonia to a substrate. So this would be a lyase type of enzyme. Okay, and so this would be, yeah, so we could see that one would be adding an ammonia to a substrate. So that would be the ammonase. Combining two molecules to make a new compound, that would be a synthetase. I don't know why I'm doing these in reverse order. Removes two H atoms to form a double bond. That would be the dehydrogenase, which either, and again, it kind of drives me nuts that this is how they name these enzymes, but these are enzymes that either add two hydrogens or remove two hydrogens from a substrate. So a dehydrogenase would be either add or remove. In this case, I guess the example is to remove two hydrogens. And lastly, to convert a cis fatty acid to a trans fatty acid. Cis and trans are names for different isomers. So this would be an isomerase reaction. Okay.